Good morning. Good morning. Good morning. Good morning. Welcome to the first of three different shared lecture times where we're going to be talking about sensory systems. We're going to start out with photoreception on Monday and Wednesday next week. We're going to deal with chemoreception and mechanoreception, respectively. So it's a bit of a change from the taste, but it's another kind of a chapter of thinking about unpacking the vast diversity of strategies that the invertebrates have come up with when being presented with the abiotic and biotic stresses of our world. We wrapped up on Monday thinking about the feeding. And we talked about parasitism and about the myriad of kind of ways that an invertebrate organism can be parasitic. Sorry, I look down, I see the chat. I see someone's having trouble hearing me. Can anyone else hear me? Could you throw that in the chat? Can you hear me? You can. There are some people that can. Okay. Alrighty. Okay, so it doesn't look like the problem's on this end, but let me, in the meantime, continue. And we will wish you good luck with solving that. Yeah, I think rejoining is a good idea. We're recording. So where are we going today? We're going to talk about just environmental cues in general, about sensing and modality, the modality of that. And then we'll talk about some classic and not so classic examples. We'll start with some cnidarian examples. We'll move into some arthropod examples, and then we'll finish with some molluscan examples. And what are my hopes for us out of this is that you are able and equipped to go out into the world to appraise, synthesize, and interpret kind of examples of photoreception, of different kind of photoreceptors, and how invertebrates use them to interact with their environment, and how that might change across different environmental gradients, whether those are terrestrial, aquatic, or marine. So when we're talking about sensory systems, we're talking about the capacity for an organism to receive cues from the environment and from other organisms. And that's a kind of a critical, essential strategy for survival. And certainly it influences behavior. Now, in particular, when we're talking about where and why, I mean, so you're looking at the banded, the ring snail there, Sepia nemoralis, and the kind of eye spots at the end of these tentacle arms. So they have optic nerves that lead into an array, a pit, and a pit is going to be a big theme for the next few days, of pigment cells that are connected to these photoreceptor cells that are connected directly to the nervous system. And this provides cues for the organism to seek food, to assess threats, to locate suitable habitat for where to live, and how to connect with conspecifics to do all those things that we talked about in the life history chapter in terms of metabolizing or reproducing. Now there is, as some of you are aware, because of different sensory modalities that you have, there's a continual bombardment of signals. And if you couldn't mute some of them or respond appropriately to them, they would be overwhelming. So there's a need to sense and respond appropriately and to be able to calibrate them, to either amplify them or to dampen them. So when we're talking about sensing and responding, these sense organs, so here again is another simple eye in a mollusk in this case, where we have a pit of cilia and photoreceptory cells that contain, are lined with pigment, this photosensory pigment. We'll unpack all of these different terms as we're going along. But that pit is then connected through each one of these cells to the nervous system. So the sense organs and the receptors and that opening of the optic cup into the pit, that's obtaining external information, shadows, images, that kind of thing. But the transition then is from external stimulus to internal stimulus, from, in this case, electromagnetic radiation, light, writ large, to chemical information, to your nervous system. So they convert light or chemical or mechanical energy when we talk about them next week, into electrical energy and sensory neurons. And those travel along, this is my traveling electrical impulse along the nervous system, into some kind of a brain, like Buck 65 was talking about in the song at the start, that the zombies need to feed upon, where the behavioral response is stimulated. So any kind, for the next kind of 3 lectures, the next chapter of sensory reception, any kind or every kind of receptor cell is highly selective for a specific kind of energy or a specific kind of stimulus. And so they're sensitive to that stimulus, and then they can amplify. There's various methods that they have of amplifying that signal. So we have, maybe it's electromagnetic radiation and some kind of pheromone and some kind of sound, some kind of a knocking, some kind of a physical transmission. Those are received by in this elegant biological, not reductionist at all, box here as selective receptor structure, a pigment cup, a sensilla, kind of different overlapping gaps in your chitinous exoskeleton that are kind of wrapped in a receptor cell that then can be amplified intracellularly and transmitted on into a neuronal signal to the central nervous system. So when we talk about receptors, as we've done, we've talked about photo, chemo, and mechano. That's kind of the way the next slices of the course are divided. But those are spoken of in common parlance often as these modalities, that we have vision. That's pretty direct translation to photoreception. Here comes a cough. My apologies. But I was saving the mechanoreceptors in your ears from the modality of hearing that cough. It touches the same kind of modality. That's also a mechanoreceptor. And then smell and taste, olfaction and gustation, or the gustatory and the olfactory, those are chemoreceptors. So chemo and mechano is next week, and photoreception is today. All right, here's the first appearance of the wonderful caramel. I'm hearing that it's staticky, and wondering if you can hear. Those of you who are having problems hearing, can you hear now? And is it still staticky? It's better. Super. Amazing. We can all help. Amazing. It's kind of a great sign for the sensory environment, the first of the sensory systems lectures, when in fact I can transmit visually and auditorily across this crazy system. So photoreception. So that's kind of in general sensory reception. But what's today's slice? The tranche today is photoreception and the myriad of strategies that invertebrates have across that done-it-all phylogeny, the map of life that we have, to intersect with this particular slice of electromagnetic radiation. Now, when we think about, and you know this from your training as a biologist, your training as a physicist, your training just as a student of science or as a citizen of the world, that when we talk about visible light, we're talking about it's a bias, right? And it's a bias that we've spoken of before when we use the phrase to see, meaning to understand. This is actually writ large. This is what we can see. So this is potentially what the realm of the world that we don't understand, or that we can't infer and that don't make common sense to us, okay? So our tranche of this electromagnetic spectrum is from just under 400 to just over 700 nanometers long. And we've got the colors that we receive, that we perceive in there across that spectrum. But of course, in smaller than in that we have infrared, and we have ultraviolet. And that many of the taxa that you're going to be thinking about, or that you already are biased towards thinking about because you were an enthusiast of the invertebrate world, they can detect more than this tranche that we can, more than this slice that we can. And in fact, there's 96% of all species can discriminate some direction of light within a few degrees. And that speaks to its importance in terms of its importance that extends much earlier evolutionarily and much more broadly than thinking about a lens forming our kind of camera eye. Again, that's our bias, and you have been exposed to kind of the wonder that we have to seeing the eye of the cephalopod. And I'm not going to talk about that a lot today because you've been exposed to it many times, and I think we should use this shared opportunity to amplify the signal of the underrepresented. But I just wanted to, on these two graphs, so the top graph on the left is to think about the light environment that life has evolved in. And the bottom is marine, that's our home environment for life. And thinking about the irradiance that, so we've got irradiance on the y-axis, we've got wavelengths of that visible spectrum on the x-axis, and then we've got different plots for how deep you are or where you live in a slice of the marine environment. And you can see that if you're if you're the Newston, you're kind of right up at the top, and you are seeing, essentially, you're exposed to that same kind of sensory environment that we are. But the deeper you go, for instance, the more important the smaller wavelengths, the shorter wavelengths. So if we go back up, so the blues and the purples become the deeper you are. And then if you're deeper than 500 meters, it's just very, very dark. Whereas in the terrestrial environment, it's not so much about where you sit in a vertical column, it's where the sun, where our star that orients us all is sitting in the sky. Whether it's right above us, whether it's at the horizon, that slices up a lot of the light that we can see. The nocturnal environment, of course, especially with a full moon, we have the reflected light of the sun off of the moon, and there is starlight as well. Both of those have to do with amplifying the importance of longer wavelengths. So if we go back again to the reds and the importance of these colors, if you're a nocturnal organism. And then we have the yellow line, like what's going on as we change. Like in the Anthropocene, during the climate crisis, we think about temperature, we think about precipitation changes. But one of the other things that is characterizing the Anthropocene is the rampant urbanization. And there's all kinds of different pollution that we think about, and light is one of the pollutants that we produce. Okay, so there is the kind of the environmental setting for a lot of the organisms that we're dealing with. But let's go back to translating that actual kind of magical act of translating electromagnetic waves in the environment into chemical energy in your nervous system. And that is happening because of these opsins, these proteins that are contained within chromatophores that sit on the cell membrane. And this is as we talk about the structure of eyes, of photoreceptors, they can be grouped, they can be situated behind a lens to focus things on, they can be sitting in a cup to focus things on, or they can just be sitting in a cell membrane where one part of it is poking above into the outside environment and one part is poking below into the internal environment. And that when light strikes that cell membrane that contains the chromophore, it alters the opsin to transmit this cellular signal and transmit it into the nervous system. So opsins in general, there's a number of them, but they are the key protein that converts electromagnetic energy into chemical energy. And they might have a single origin within the eukaryotes, and then a different origin independently in the bacteria. And then since that metazoan event, there has been a diversification into a number of different mechanisms, but the underlying machinery, the opsin structures, is remarkably similar. So the structures, this is so to orient you, we've got kind of an old school Darwinian style phylogeny here from metazoans, so eukaryotic animals, a dun phylogeny, we've got cnidarians that branch off into the bilateria, and then we've got annelids, arthropods, deuterostomes, with some different examples of taxa in cartoon form. So the lancet, Amphioxus, some different mollusks, annelids, cnidarians, our own lineage that we'll ignore over here. But the other element that's cartooned out here is that photoreceptive eyes, they're kind of two types. There's a ciliary type, so that is in pink. So you see these like little fingers kind of emerging here from the scallop, or this almost like an old school TV antenna here in the box jellyfish, the cubozoan. These are all ciliary type photoreceptive cells. So they have a membrane that develops from a basal body and then a cilium. So literally like a cilium that protrudes into this photoreceptive area. And they're used generally as simple light detectors, and we have them in our cones for bright light vision. Now the blue structures, these are R-type over rhabdomeric, where there's microvilli that are contained. So we've got little tiny, so almost like this little toothbrush here with the Drosophila, or the double-sided toothbrush over here for the squid, or the, I'm not sure, but that makes me think of The Simpsons with the horseshoe crab here. So that pigment-containing membrane is formed with microvilli, and these are single photon detectors, and they're excellent for lower levels of light. But you can see that their distribution, even in this cartoon fashion, across the animal phyla, our map of life, they're broad, they're widespread, these two kind of broad strategies. And so when we're thinking about invertebrate eyes, whether we're thinking about pigment cups or optic nerves, so like from the pigment cups on the back of or at the anterior end of your platyhelminth flatworm to the dissected optical nerve and like image forming camera-like lens of the hunting squid, that we have a vast array of intermediate stages here. So we have simple eyes. 85% of invertebrate phyla, of animal phyla, have some kind of pit eye. We've got spherical lenses. We've got cases with multiple lenses, so amplifying that internal, that signal as we transmit from EMR to neuronal to the nervous system. We've got reflector eyes. We'll talk about scallops later on, again with multiple lenses and reflections. And then we have the evolution of the compound eye. And we'll spend a bunch of time today thinking about the arthropod or the hexapod compound eye. And we have the evolution of apposition eyes that we see in arthropods. We see refracting superposition eyes, reflecting, and we'll unpack some of these words. But the compound eye can occur in so the compound eye itself, you're picturing the housefly, there's a multitude of forms that particular evolutionary divergence has followed as well. So we have eye spots from amongst the metazoans or even some of the protozoans to so planarians and annelids. We have single-chambered eyes. We have compound eyes. We have photoreceptors with microvilli, the rhabdomic ones. So cephalopods, snails, insect larvae with mirrors or lenses, and then compound eyes is with lenses or with mirrors. So lenses being like the adult insect example, and with mirrors, the shrimp or some of the marine examples. So let's think about eye spots for a second. So we talked about that very simplified diagram of the internal membrane, so the membrane with a chromophore and poking into the outside and retained in the inside of the body. But let's talk about this kind of diagram from your text that's thinking about a very simple eye spot, where the photoreceptors, instead of being distributed across the body, become concentrated. This also tends to happen around edges or around in the bilateria when there's a directionality, when there's a head or some bit of cephalization. So they can sense ambient brightness, but they're insufficient for vision. So we have, in this case, so not with a bilateria, but with a hydrozoan medusa here, we have the underlying gastrodermis, and then on the outside, in the epidermis, we see these pigment cells that are arranged around a lens, and the pigment cells are connected directly to neurosensory cells, to nerve cells. Now, from this point, in cnidarian evolution, from the eye spot, there's actually been a tremendous diversification of how jellies see the world. So jellies being perhaps some of the first organisms that any alien visitor to our planet, like if you were to look at Pixar's Elio and Aurelio and think about where that alien's going to bump into, it's our planet Earth is covered in the sea, and so they're going to bump into the sea, and more than likely, if they bump into the sea, they're going to bump into a jelly, and these will be some of the first bits of eyes that see that alien life. We've got eye spots. We've got Cnidaria with eye spots, with everted pigment cups, that have lenses. That's kind of like what we just saw. And then we have an actual lensed eye. And each of these jellyfish eyes has different ultrastructural details. So they're derived from different cell types, and they're derived differently also in an evolutionary perspective. That what we see across Cnidarian life is that multiple times we've seen the appearance of eyes. And these known morphological differences, where you're looking at eye spots here, or pigment cups, or actual lensed eyes like on the corners of the cubozoan, that this is consistent with an interpretation of conversion evolution with multiple origins. And this is supported from a physiological or a biochemical analysis. So this is our phylogenetic analysis. But if we overlay with that, an examination of the underpinning chemistry of how these eyes work, the photosensitive proteins are also different and support this independent evolution. So that's our kind of focus for a second on the Cnidarian, the diversity of strategies within the Cnidaria. And I said I wouldn't talk at all about the camera eye, but I mean, of course, you've seen it before, you've seen it in your text, you've seen it in lots of your training or your enthusiasms as an invertebrate biologist. But if we compare a cross-section of our own eye here on the left and a cross-section of an octopus eye, we see incredible similarities, that we see a lens and a cornea, an iris opening that provide really true and vibrant imaging on the photoreceptive cells and into the nervous system on the back of that eyeball, that can shape sense, that can fine-tune directions. And there's more diversity of invertebrate strategies to talk to, so I'm going to leave that one there. Caramel's going to remind me to check in, and if you're all still hearing and seeing me, then we're good to keep going. Now, if we move from the mollusk the quick hit of the squid or the octopus and the thinking about jellies across the world, if we move into the terrestrial environment right now to think about compound eyes, in this case amongst hexapods, and whether we're in a terrestrial environment like this stonefly here, or an aquatic environment in the water like this Daphnia. So we see simple pigment cups, and we see ocelli. So there's like a light spotting cell there, but you see very simple eyes here. So with the stonefly on the right-hand side, the yellow insect, this is the compound eye that my mouse is tracing over. The ocelli are these 3 dots in the center, in the forehead. And you've seen them in many insect groups, but these are just simple pigment cups that are kind of light-dark receptors. This is not an image-forming kind of a structure. But you'll see its importance we can infer from the fact that it's retained in many, many lineages across the diversity of the arthropods and across the hexapods. But let's kind of walk into the diversity of the compound eye, seen here with this kind of side view at this gorgeous compound eye of an undescribed species of staphylinidae rove beetle from Costa Rica, where I work. And so you see this cluster of Ommatidium. So there's many Ommatidia. So this is an Ommatidium. The compound eye contains many Ommatidia. And each Ommatidium is a cluster of photoreceptors that is surrounded by support cells and pigment cells. And when you think about, and this is one of the kind of themes that I'm going to ask you to do, is to empathize, is to develop empathy with those that see the world in a very different way that we do, is to think about how this compound eye provides a very different view of the world than our own camera eye does. So the resolution here for the compound eye is determined by the number and the size and also the orientation and also the photoreceptors, but writ large. The resolution is determined by the number and the size. So the fewer that you have, low resolution. The sensory processing is not done in the organ, it's done in the central nervous system. So some of the pluses of having this compound eye, you can see this is a different species of beetle. You can see the shape of the ommatidia are different, that the space between them are different, so that the angle that is achieved between each ommatidia or each ommatidium is different, and all these have to do with sensitivity and acuity. But it provides the capacity for the organism to look in several directions at once. And each ommatidium provides the brain with a one-picture element, so it's like a pixel essentially. And if you wanted to think about it it's not a whole bunch of different images. But I'll come back to this. Let me kind of challenge you again, since we've been talking about these staphylinids. These are two different species of staphylinid. One in the cold, wet cloud forest in Costa Rica with very flattened eyes, very hexagonal, almost like honeycomb kind of shape. And then in the wet but much brighter environment of the rainforest, you see an organism that has an eye that is protruding out from the body, that has these microtrich hairs and a much more densely arrayed, kind of protruding, almost like you've been baking tiny loaves of bread, and they're all poking up over the edge of the pan here. And think about the kind of light environment that these live in, so much darker on the left, slightly, still moderately dark, but occasionally much brighter here on the right, and or the kind of life that they live. Are they predatory? Are they active and moving? Are they scavenging? Are they detritivores? What role does light reception play? Is it just predator avoidance, or is it prey tracking down? So when we think about arthropod eyes, most of them, like this, so this is a hexapod writ large. This is a colembla from the same forest as the staphylinid was at the last. But they have these two types of eyes, as you saw in the stonefly several slides ago. They have this simple ocelli, and they have the compound eye. The compound eyes are generally on the lateral and the side of the head. They form images. They let you detect edges of things. They're often, for this, because of the physics of how light is being transmitted through those ommatidia, they're often not symmetrical, that we see different regions of the eye where the ommatidia are shaped differently, individually and in packed together in relation to each other. And this has to do with being able to use different parts of this massive organ to focus on, to use it for different things. And in fact, as you'll see, some of them have different sensitivities to different portions of that electromagnetic spectrum. So the simple eyes are great for detecting low light levels, and together they provide a very fast response time. And when we think about stalks here, so we have eyes that are stalked. They're not sessile. They're not trapped against the head as the colembla or the staphylinid example. But these dipteran examples are kind of stuck out on, and you can get kind of crazy examples of these compound eyes that are out here, looking like a little barbell or a dumbbell. So multiple ommatidia that can be sessile against the body or stalked against, but the focus of the lens can't be varied, and this is part of the reason why the different regions of the eyes have different jobs. So there's a wide range in the number of the ommatidia. They can go from dozens, 5, to literally tens of thousands. And most of them are sensitive to ultraviolet, so like shorter than what we're seeing, so beneath blue, so 300 to 400. And phototaxis, so your orientation towards light, is often cued not by the parts of the way that we view the world, but are cued by ultraviolet or polarized light rather than visible wavelengths. And I love this. So this is I might have shown you this. I love this picture. This is an undescribed species of fly that lives in the leaf litter, crawls around in the leaf litter in Costa Rica, but occasionally this male, and I know it's a male, I'll tell you how in a second, emerges from the leaf litter and dances, kind of competes for female attention with a vertical dance that they do in the air column just above the leaf litter. And the reason I know this is the male that does that is because the size, see that line right here that my cursor is moving across? That's essentially the same line as you would see in an old person's glasses that have different focal distances. So this allows the arrangement of the ommatidia in two different kind of bands, in this case, allows great depth perception, motion reception, and so allows them to be very precise dancers for the female. There you see this tiny little muppet sticking out in the center here. That is the ocelli, so it's still just simple. They're simple. But let's look at this old school diagram of the hexapod eye. I love this one from the early 90s that's looking at a fly eye. So this eyeball is sitting somewhere very close to you on a windowsill. And if you wanted to take your the microscope that you have in your pocket, your phone, and zoom in and zoom in and zoom in on that fly eye, you'd see the individual ommatidia contained within the fly's compound eye. And I just wanted to draw attention to the fact that different slices of each of the eyes, so different lateral slices, are receptive to different arrangement, different wavelengths of light. And so there's a highly complex arrangement of the receptor cells and of the opsins inside of them that are across that eyeball. Let's dive in a bit and look at this, the compound eye in cross-section. So we'll still think about kind of these big-headed, we'll think about fly examples often, because flies have some incredible eyes. So here we have a compound eye that we've taken a slice out of, and we take a look at this one where my cursor is moving across, one ommatidium. So each ommatidium there's a cornea, there's a crystalline cone, there's some iris or a pigment cell, and then there's a long column of receptor cells that have these rhabdomes, the rhabdomes that contain the pigments, that are connected to a basement membrane, and through the basement membrane to the receptor cells or the nervous system. So each ommatidium has a lens, a cone, light-sensitive visual cells, and pigment cells. Let's blow this up a bit. And so there we've got our cornea. So there's the scale that we're talking about. So this is a big eye. So it's a very small cornea with a cone that leads through the pigment cells and into the rhabdome. Now, so each one of those ommatidia is focusing light and has pigments for detecting color, for detecting light. That image is projected right side up. And so images or insects with large globular eyes, like this fly here on the bottom right, has essentially a 360-degree panoramic view of the world. So they don't have to turn their head to get a sense of their environment. Now the rhabdome in the center here, almost like the little backbone down this ommatidium, this is where the mass of microvilli, so that little kind of in the blue and pink phylogeny at the top, this is the one that I call a toothbrush, that belong to the cells of the retina, and they have the highest density of the opsins, and in this case, in the hexapod example, rhodopsins. Let's take a slice, a cross-section through here, through halfway down into the rhabdome, and you can see so this is looking at those kind of clusters on the outside. That's the ommatidium, that's the edge of the ommatidium. And then in the center, we've got the reticular cells that are comprised of these microvilli that contain all of the density of opsins. Now, that's kind of one ommatidium pulled out and examined. Now, how they are arrayed in the compound cell changes, and it changes largely dependent on what kind of light environment that insect lineage has evolved within. So for species that live operate nocturnally or live in low light, like dense forests where the much of the light above the canopy doesn't go into the subcanopy environment, we see superposition compound eyes. This is a word that I promised earlier that I would unpack. So optical superposition occurs when light is focused by the cones across a fairly large clear zone into the photoreceptive cells. So it's focused, and it helps concentrate the light from more than one ommatidium into an axon, into a single axon. Now, an apposition compound eye, which is on the bottom here now, there's a much smaller space in between the rhabdomes and the cones, and the light that's coming through one ommatidium is entering one cell, and it's confined to that cell. And we see this in kind of out in the open field in a high light environment, in insects or hexapods in a high light environment. So the compound dye, whether it's superposed or apposed, comprised of many individual Ommatidium, and I mentioned earlier that can extend across a range. That Colembala example, the purple freckled one from Costa Rican rainforest, cloud forest in fact, and I think 5 to 10 Ommatidia. This dragonfly from outside of Algonquin Park could have 28,000 different Ommatidia. Now, the arrangement of the Ommatidia, now we talked about their superposition, the apposition, how the space in between the rhabdome and the cones can deal with amplifying, concentrating light in a low light environment. But there's all sorts of other variables that evolution has kind of played with in changing the receptivity of this kind of pixelated screen. So we can change. So consider here, there's this kind of image you're going to see for a couple of slides here, but consider that we've got two different eye sizes with two different radius. That's R1 and R2 here. So we've got a great big eye, the white one on the outside, and we've got a much smaller eye here on the inside. The facet diameter, that's the diameter of the Ommatidia on the outside of the eye. So increasing, and we'll talk about kind of changing, and then the angle between, that's this variable here, that's the angle between. So how across the eye shape, how having a great big eye would change and make a wider angle than having a very small Ommatidium and a much tighter angle or a closer angle. So increasing the diameter of the Ommatidia should increase the number of photons that you catch and should improve sensitivity. So maybe you would expect to see, you can think about the kinds of environments where you expect to see large facets, large diameter Ommatidia. But it is invertebrates or hexapods writ large, whoever's got these compound eyes in the terrestrial or the marine environment, there's kind of checks and balances or costs and benefits towards changing different aspects of this. That, for instance, if you make a great big diameter and neither the shape of the eye changes nor the size of it, so its radius changes, that's going to reduce the number of Ommatidia. So if you've got a constantly held eye that can have many small Ommatidia or fewer large Ommatidia, fewer large Ommatidia increases sensitivity, lets you capture lots of different light, but it increases this angle between the individual Ommatidia. And the angle is kind of our measure of how fast, how rapidly the eye is able to sample that environment, and it decreases the acuity. So the acuity being like the how sensitive you are to the edges of things. So alternatively, an organism could increase the eye radius. So you could think about a smaller eye and a bigger eye. That should decrease the interangle. That should increase acuity, so you can see much more of the fine detail, but without changing the size or the shape of the Ommatidium or the surface area of the eye or the shape of the eye, then the visual field is going to change. And so amongst arthropods, the relationships between the size of the eye, the shape of the eye, the size of the Ommatidium, and then the angle in between those Ommatidia leads to trade-offs between sensitivity to any light at all and acuity. So like what are you going to do? What's the high quality? Do you want just all of the information from a dilute environment, or do you want the most finely tuned information? Now I'm going to ask you to think about that graphically and find, if you could, your annotation tools. Because I'm going to array here some faint axes, but we've got across the bottom an axis that could be urbanization. It could be just from forest interior to edge, like a forest field environment, where we've got temperatures. The numbers on the bottom are kind of ambient temperatures, from moderate temperatures to very hot temperatures, but essentially it's brighter. It's brighter across the x-axis. And then we've got the size of the Ommatidia. So there's a concentration gradient on the x from like deep cool forest to hot bright field. And what I want you to do, just based on what we've been thinking about so far this morning, is to grab your annotation tool and show me the kind of relationship you would expect to see between the size of the eye and that gradient on the x-axis. just going to say, as you're sketching these things, if you have a second thought, don't erase your first one. Keep them up there. This is great. So we've got multiple thoughts that are appearing. And if you've got multiple thoughts in your head, that's kind of a natural human state. Share with us all of them. Okay, so... And you don't have to be concerned about your lines, because I have a lot of things on my desk underneath the screen here, and none of them are rulers. All right. So, thank you. Well done. We've got two different hypotheses here. We've got the hypothesis that as you move from the interior of the forest, a cool, dark forest, out into the field, that the eye diameter is going to get bigger. The ommatidium is going to get bigger, or that they're going to get smaller. Let's take a look at what the data actually show. And so for a set of bees, this is the trend that we actually see. So darker areas having larger eyes, larger ommatidia, that they're sacrificing the acuity. If the eye shape doesn't change across this gradient, they're sacrificing the acuity, the fine detail, for harvesting more photons in a photon-poor environment. So, very cool. Well done, all of you. Now, when we think about acuity, the detail that can be seen, the greater detail, the greater the acuity, this is, so decreasing, getting smaller ommatidia, that increases the acuity. But generally, if you were to think about acuity here on the y-axis and think about the diameter of the eye, we can plot several different kind of characteristic invertebrates across the small end of having very small eyes. But you see, if we kind of throw in some vertebrates just for giggles, you can see, and then we put as these researchers did, kind of put a bracket of where legal blindness occurs for our own species. You can see that most of the invertebrate world that's captured in this, mostly these are hexapods, but we have some mollusks as well, are legally blind, according to us. And that's just based on the acuity. That's not great. And so so that's not a cost, but there's no, the benefit of acuity that we enjoy, they don't. What do they receive? What selective advantage might they have for this kind of a compound eye? One of them may be, remember, of course, evolution is not working away at the vast diversity of potentials, engineering potentials of life to come up with the best solution. Evolution is always satisfied with it's good enough. It's better than anything else. It's good enough. And so the compound eye is not by any way or perspective the best eye. And some researchers think it's like kind of a bad solution, but it's clearly been good enough to support the diversity. One of the ways that it exceeds expectations, that it does better than is in a response to motion. It may be, so this is come with me over to the left-hand side here, to the graph. This is a look, so through a predatory dragonfly, many ommatidia, tens of thousands of ommatidia. When the prey item against a bright background is very close, so that's this slice, this kind of decreasing or increasing slice of gray. When it's very close, it can respond. This is kind of our interpretation of what it's seeing through there. And the farther away that it gets, the more chunky it gets, but it still can respond in this modular view very sensitively to motion. And so if we take a look in another way of looking at this, again, we've got the motion on the left hand from top to bottom through time. You can see the brown fly kind of moving around, flitting around in this very complicated, cluttered environment. And then if we pixelate it to what we imagine and kind of take some of the contrast out, as the compound eye would be able to do, you can see that in a very complicated environment, this may facilitate the predator towards seeing that motion and responding to it in a very otherwise cluttered world. Cluttered especially if you don't have, you're seeing what I'm seeing right now, which is not very acute at all. Now, because there's all sorts of fantastic predatory examples. This is a robber fly. This one on the right-hand side, this is in Costa Rica. This is a fly sitting on an acacia plant, but you can see these stalked eyes. This is a highly mobile predator, and it's used in vision studies or in photoreceptor studies or in predatory studies a lot because, let me go back, in the blink of an eye, these robber flies can, with incredible position and speed, move out and hawk. So from a roosting position out into the environment, capture the prey and return to its resting position. They can capture this prey incredibly quickly and incredibly accurately. And both neurologists and photoreceptor or kind of light biologists have always been intrigued by this specificity because these flies maintain what's called a constant bearing angle, where they proactively change their angle not on the initial situation where they departed their perch, but they alter it based on the speed. They lock onto that speed and then alter, so they maintain this constant angle across time to make sure they intercept it. And they're doing this with a really small brain. And this is the same kind of activity that your dog is doing when it's catching its frisbee in the air, or your cat is doing it when it's catching a bird or a little thing that you've flipped up into the air. And so it's kind of amazing to us, again, that this animal with a very small brain is using the same strategy. And so the acuity may be compromised. Here again, so we've got robber flies very close. They can see quite clearly. And then as you move farther and farther away, they become dimmer and dimmer. But this is kind of the high end of the highly visual predator. So even 3 meters away, they're still seeing. That's kind of at least as good as what I would see without my glasses. The water flea, the springtail, they don't see much, even when things are very, very close. Now, what they're seeing, of course, is varied by the kinds of opsons. We have 3 that lets us see millions of colors. So if you increase the number of opsons, you have an increased perception of color. And I love the fact to emphasize at the end here that some arthropods have more than 30 opsons, so they can see colors that we can't even imagine. Now, the next couple of minutes, I'm going to move into revisiting some molluscan examples because the compound eye is you could have a course on an arthropod compound eye, but you could also have a course on the incredible diversity of molluscan responses to the world. One of them here is the fact that scallops have eyes in their mantle, right? Remember the mantle of the mollusk? It can practically become anything. And one of the things that it can become are these eyes that have mirrors in them that are arrayed around the edge of the opening of the 2 valves. And that they have multiple retinas, which lets us think, so this is our eye here. This is the scallop eye, that they have multiple ones around the edge of the mantle with 2 retinas that we think allow them to perceive the periphery and to focus clearly and to be able to respond then to their environment. So it's passing through a very complicated eye that's made from the mantle, the thing that's making the shell or as a foot or all these other things. But the other cool thing about it is that it's passing through a mirror. Again, this is another way of amplifying the signal. And the mirror is made of crystals of guanine that are at the back of the eye. So this is their crystals of guanine back here, the purple. So it's structured less like a camera as our own eye or the other molluscan lineage, the octopuses, but more like a telescope. And they can open them, they can contract them. So the mantle providing eyes for mollusks. But then what about, you remember the polyplacophorans, another lineage of mollusks, where they make their eyes out of their shell. Right? So that the way that the grains are laid down in the protective part of the shell are haphazard. Light doesn't pass through them. They're structurally very strong. But in the lens, so we have a lens and a cornea, then the lens is made of argonite. This is literally part of the shell, but it's connected to retinal cells that lead to an optic channel, and the grains in that part of the shell are not arrayed like plywood. They're aligned to let light pass through them. So that provides areas that are not great at repelling a load. Like if you're to step on the eye will crack. But they're protected on the edge by these non-sensory, these solid protrusions, this kind of shell-like, the shell-like part of the shell. So simultaneously then, the chitin shell, the polyplacophorine shell, is seeing the world, multiple eyes being formed all the time around the edges of the shell, not in the mantle like the scallop, but in the shell, and also providing mechanical protection. Incredible. All right. And that's the time that we've got. So, like so many items in this course, this is our kind of dipping our toe into this. And if you're interested in this kind of biology, there's all sorts of different aspects, phylogenetically or mechanistically, that you can explore in and around your University of Guelph. And where are we going? We're going to talk about chemoreception next week. You're going to be talking about sensing and responding in the lab next week. Your peer review is due this Friday. Your case study is accessible as of Thursday, per usual. If you haven't already started your invertebrate R, the second assignment, I would urge you to do, because November 7th is not getting farther away. It's getting closer. So I'm going to stop recording here in a second, and hang around if there's any questions. And other than that, I'm going to encourage you to be good to yourself and be good to others, and look forward to seeing you again next week.